Happy Halloween, everyone. Thank you for being here. And today it is my pleasure to share with you our work on developing molecular contrast agents for liquid biopsy together with Shervin. So, what are liquid biopsies? Liquid biopsies refer to these analysis of tumor derived components in bodily fluids, including blood, for diagnostic purposes. And specifically, one analyte of great interest is cell free DNA that consists of fragmented DNA molecules that are released into the bloodstream by a variety of mechanisms shown here on the left. And specifically, the majority is thought to really come from apoptosis. And in healthy individuals, most of this cell free DNA comes from white blood cells. However, around 30 years ago, scientists realized that the blood of cancer patients actually also contains tumor-derived DNA fragments, and this really generated a lot of interest. And one specific term I want to discuss here is the tumor fraction, which is the fraction of tumor to total DNA in these samples. And after the serialization, there has been a whole host of genetic and non-genetic signatures that have been identified in these DNA fragments that really make them carry very rich information about clinically relevant genetic variants, tumor burden, even tissue of origin, and all of these from sort of a minimally invasive and the convenience of a blood draw. So it is for all of these reasons that ctDNA has really been coined as a very promising biomarker for oncology, and it is currently being investigated across the whole landscape of sort of the patient journey. So for instance, ctDNA tests are being investigated in early detection through pan-cancer screening tests to be able to detect cancer early and treat it when it's most curable. It's also being investigated to detect recurrence after curative intent. And to inform sort of treatment escalation for patients with minimal residual disease. And finally, also in the setting of sort of advanced cancers, when we're trying to inform therapy and tumors evolve and tissue biopsies becoming feasible in the setting of metastatic spread, it can also be helpful. And it is precisely in this setting of sort of advanced cancer that ctDNA has had the most success, with the first FDA approvals being granted in 2016 for EGFR inhibitor prescriptions. However, despite like all of these, exciting prospects, the reality is that the sensitivity really is not where we want it to be for broad clinical incorporation. And this is evident from sort of the numbers I'm highlighting here for the different applications we just discussed, where we're not really getting more than 50% sensitivity in a lot of these settings. So we asked, why are we hitting this sensitivity limit? And although there are very many technical challenges with sort of detecting these DNA molecules in blood, the more fundamental limitation is that most blood samples just don't have enough ctDNA molecules in them. There's just very low levels in circulation. So to illustrate these points, a typical blood sample only contains around 20 nanograms of cell-free DNA, which amounts to around 3,000 diploid genomes. So what these means is that for patients with relatively high tumor fractions, down to 0.1%, we're at odds really of finding any one mutation and sort of calling this blood samples cancer positive. But as soon as we sort of go lower in terms of tumor fraction, our probability of really coming across any one mutation is really, really low. And unfortunately, for a lot of the patients we care about and patients with microscopic disease, we're really at very low levels in terms of tumor fraction, which makes it really challenging to deploy this clinically. So we turned to biology to try to understand why cell-free DNA levels were so low. And what we learned from reported literature is that billions of cells are dying in our body every day, and they're shedding their material into circulation. However, the bottleneck is that it's very rapidly cleared from circulation. And there's really two major mechanisms for ctDNA clearance. The first one is cellular uptake, and the second one is nucleus digestion. So as I mentioned in the first slide, ctDNA or cell-free DNA generally is mostly generated through apoptosis. And in this process, DNA is fragmented at vulnerable internucleosomal linker sites that results in sort of this DNA being carried wrapped around a histone core. And this mononucleosome, as we call it, is actually only 11 nanometers in diameter. So similar to many of our nanomedicines, it's readily uptaken and cleared by macrophages in our body or the MPS system. In addition, there's a lot of circulating DNases in addition to intercellular DNases that will eventually find this DNA and clear it from circulation. And both of these processes mean that the half-life of cell-free DNA is only in the order of 10 minutes, which is really, really low. And this leaves very little material for us to detect from a blood draw. So for this work, what we asked is whether we could develop agents that could slow down cell-free DNA clearance such that we would have like a buildup of material in blood and by extension be able to recover more tumor DNA for analysis. And this is where we coined the sort of term of molecular contrast agent because these agents sort of allow you to detect molecules that were otherwise invisible prior to using these agents. And today, what we will be presenting are two completely orthogonal approaches to sort of achieve this strategy. The first one will be a nanoparticle contrast agent that acts on the cells responsible for cell-free DNA clearance to achieve just this. And the second one will be a DNA binding contrast agent that protects the DNA itself from clearance to increase its recovery from circulation. So starting with our nanoparticle contrast agent, to develop this agent, we took inspiration from the field of nanomedicine, where macrophage uptake has been a delivery challenge for decades. As we're all aware, when we inject any nanoparticle into the body, a large fraction will be sequestered in the liver. And there have really been a whole host of approaches to try to mitigate this phenomenon. And what I really want you to focus on right now is this idea of Kupffer cell saturation, which essentially is the notion that you can deliver a high dose of particles that will occupy macrophages and essentially overwhelm their uptake capacity, such that when you then inject a subsequent dose of therapeutic nanoparticles, there will be less hepatic accumulation and more of them will get to your target site. So what we hypothesized for our first contrast agent is whether a competing nanoparticle that is efficiently uptaken by the MPS system would attenuate the uptake of our nanoparticulate cell-free DNA, so our endogenous nanoparticle of interest, the mononucleosome, and therefore, allow us to accumulate more in blood and detect more ctDNA molecules. To test this hypothesis, the first thing we did was go in vitro with a in vitro system where we can treat cells with liposomes and subsequently give them Cy5 labeled mononucleosomes or analyte of interest and then measure uptake. And we tested 3 different liposomes in vitro with similar size distributions but a range of surface charges. Excitingly, what we saw is that 2 of our liposome formulations, the DSP and DSPG based liposomes, had very nice dose-dependent uptake inhibition of Cy5 labeled mononucleosomes. Intriguingly, the DSPC liposome did not, but this is somewhat consistent with the literature that has found that negative charge is sort of important to increase interactions with macrophages, and this could hypothetically maybe make for better contrast agents. Excited about these results, we went in vitro in vivo with our best candidate, which was the DSP-based liposome. And we first just validated that the liposomes were going where we wanted them to go, which was the 2 main target organs for the MPS system, the spleen and the liver, which was the case as shown here by biodistribution of liposomes at 1 hour. And we then asked the question of whether these actually decrease the clearance of cell-free DNA, which was what we had hypothesized. To do this, we used a healthy mouse. We injected them with liposomes or PBS and subsequently administered mononucleosomes carrying a known DNA sequence that we could track by qPCR and performed longitudinal blood sampling after that. And consistent with our hypothesis, what we observed is that at 1 hour, we increase the amount of mononucleosomes we were recovering or DNA that we were recovering, 1 to 2 orders of magnitude with liposome treatment, and this increase was significant. And this was also consistent with increases in the half-life of this cell-free DNA in circulation following treatment with either the 100 mg per kg or the 300 mg per kg liposome doses. However, all of this is really using an exogenous cell-free DNA source. We were injecting this mouse with synthetic DNA material. So we next wanted to see whether this had actual effects on real endogenous cell-free DNA levels. To test this, we turned to a tumor model of cancer, specifically a colon carcinoma flank tumor model. And we chose this model because it had previously been reported to have many other genomic alterations that we could track in plasma. And additionally, there had been papers showing that this model had this ctDNA was diagnostic in this model for late stage tumors. This is important because there aren't that many preclinical models out there for looking at ctDNA in mice. Most research is really done in humans. So we were sort of trying to figure out what the best model would be. So we inoculated these mice, waited until tumors grew out for around 2 weeks, and then treated our mice with liposomes or PBS and sampled blood 1 hour later. And what I'm showing here on the right is actually that post 1 hour post treatment cell-free DNA yields, and this is now endogenous cell-free DNA levels, indeed follow the same trend that we saw for the exogenous material. So we're recovering more endogenous DNA with liposome treatment as we were hoping. However, this is just cell-free DNA levels. So we next designed a panel or a test that allowed us to very specifically find tumor mutations in DNA to be able to detect this tumor-derived material. And this assay is based on a hybrid capture where we can very specifically look for loci in the mouse genome that correspond to mutations in the cell line and quantify both tumor DNA and also tumor fractions in our samples. Using this assay, we were able to show that pre-treatment, so prior to any liposome administration, we have comparable number of mutant molecules or mutations that we're able to detect in these samples. However, 1 hour after treatment, we again, are able to recover up to, in this model, 10-fold more mutant molecules with liposomes. In addition, given that we have this probe panel, we're able to show how liposomes affected the fingerprinting or mutational fingerprinting of this model. So here, these heat maps represent, each vertical line represents a mutation in our panel. And what we were able to show is that pre-treatment, there's really only a handful of mutations we can detect. But 1 hour post-treatment, we're really seeing a lot more mutations, as shown on the lower row. And this is important for genotyping or for forming therapy selection. Finally, although this flank tumor model was kind of useful to investigate the effect of liposomes on cell-free and ctDNA recovery, it had one main limitation, and that was that tumor burden was not really proportional to ctDNA shedding. And this is what we typically see in humans and what we would need to really show that our treatment could improve the sensitivity for detecting tumors and allow for detection of smaller tumors. So to do that, we turned to another tumor model. This is now a lung metastasis model where we inject a luciferase version of the cell line intravenously. And we found that this model had a more physiologic shedding in that more burden amounted to more ctDNA being shed. And we treated mice with liposomes throughout tumor progression to see how sensitivity changed at different stages of tumor burden. And excitingly, what we were able to show is that across really all stages of tumor progression, liposome treatment, here shown in orange, improved our ability to detect mutations in this model relative to the blue PBS control. And this improvement was up to 60-fold in our latest time point, the 3rd week time point. In addition, by using a simple classification metric, which was like if you are able to detect more than 5 unique mutations, you classify a sample as ctDNA positive and otherwise as ctDNA negative. What we were able to show here depicted as sort of crosses for negative, like no cancer detected, and circles for yes, cancer was detected. We were able to observe that a lot of our liposome treated samples, we were able to detect cancer, whereas a lot of our PBS samples remain undetected, even at really high burdens. And this sort of resulted in higher sensitivity across all 3 weeks with our liposome treatment relative to the PBS treatment. And with that, I will hand over to Shervin for the second part.
Thank you, Carmen. So as you heard from Carmen, we've shown in this first part how by intervening on the cells that take up cell-free DNA from circulation, we can improve the recovery of cell-free DNA and circulating and the detection of circulating tumor DNA. In the second part, I'm excited to share some data with you about how we can actually intervene on the analyte itself, on cell-free DNA, and protect it from clearance via a DNA binding contrast agent. And so our idea here was based on the observation that cell-free DNA has a very short half-life in circulation. We thought, well, what if we actually couple it with a longer-lived molecule that can protect it from nuclease degradation as well as clearance from circulation, and hence allow it to build up in plasma, enabling better detection of tumor DNA. And we thought about a lot of different binders that we could use for this purpose. But for this proof of concept work, we really focused in on antibodies. And this was for a couple of reasons. One is they're already long-lived in circulation and have very well established methods of or mechanisms of recycling. And then second, there's very well-established methods for engineering and production of antibodies, and they do have a long track record already in biomedicine and clinical use. So as a first step, we decided to test just 8 different antibodies to see if we could find one that bound to double-stranded DNA. These are all antibodies that have been reported to bind to the double-stranded DNA, and we just did a ELISA here. And we found 4 that showed some evidence of binding on an ELISA at sub-nanomolar concentration. We picked one of these antibodies, 35I9, for further development. This is an antibody isolated from a lupus mouse model. It's an IgG2A isotype with a reported dissociation constant with DNA of 90 nanomolar. Now, as the first question we asked was, so how does this antibody interact with free and nucleosome-bound DNA, the components of cell-free DNA? And to do this, we did a gel shift assay where we incubated free double-stranded DNA. This is about 150 base pairs, which is around the length of cell-free DNA fragments, with various concentrations of antibody. And we showed that, indeed, as expected, we see shifted bands at higher concentrations of antibody corresponding with binding of this antibody to this free double-stranded DNA molecule. And we saw several bands, which we hypothesize are higher ratios of antibody to free double-stranded DNA. But then the next question was, well, what about nucleosome-bound DNA? So we did the same experiment, but this time we also included nucleosome-bound DNA. And so you can see evidence of that with the appearance of this middle band, which corresponds to free DNA wrapped around the histone octamer. We again saw the same bands corresponding to free DNA binding to the antibody, but we actually also saw some faint bands in between, which we hypothesized corresponded to the antibody interacting with the intact mononucleosome. And we actually confirmed this on a Western blot of the same gel for histone H3. So this together established that this antibody can indeed interact with and bind to the components of cell-free DNA, free DNA and nucleosome-bound DNA, at least in vitro. The next question was, well, what does it do on the kinetics of clearance of cell-free DNA? And to do this, we did a similar experiment to what Carmen had already described. So we injected this antibody with nucleosome-bound and free DNA, and we did serial blood samples and quantified the amount of exogenous DNA that we injected. And what we saw is, and we were excited by this, is that when you normalize to the sort of 1 minute blood draw, you actually see significantly decreased clearance of this injected DNA compared to an isotype control, as well as to just the injection of DNA alone. So this got us excited, but the excitement was short-lived because then when we actually looked at the absolute quantity of WDM601 or the exogenous double-stranded DNA, we saw no effect. So there was no difference at 60 minutes. And you can already tell based on these plots where the problem may lie. It's that at 1 minute, a large fraction of the DNA that we injected was being cleared in the presence of anti-double-stranded DNA antibody. And our hypothesis here was, well, what if FC receptors, interactions with FC receptors are leading to sequestration and clearance of the DNA that we're injecting? And to test this, we did a similar experiment where we, in addition to the anti-DNA antibody, injected FC receptor blocking antibodies. And we saw that with blocking FC receptors, we actually reversed the initial early clearance of double-stranded DNA. And excitingly, now at 60 minutes, we did see at least an order of magnitude improvement in how much DNA we were able to recover in plasma. So we were excited by this. And the next step for us was, well, let's engineer our antibody to recapitulate this effect. So instead of trying to block FC receptors with additional antibody, let's just abrogate the interaction between FC receptors and our antibody. And so we introduced a number of mutations to the FC domain of our antibody to try and disrupt this interaction. And we expressed and purified these antibodies, and then asked a similar question of how did these affect the clearance of exogenously injected double-stranded DNA. So again, we did a similar experiment where we co-injected and then sampled at 1 minute, and this time at 60 minutes, and we quantified the amount of exogenous double-stranded DNA in plasma. Similar to before, we saw pretty rapid clearance with the isotype control and some improvement when we blocked FC gamma receptors with the wild type antibody. And then among our 3 engineered variants, there was one clear winner, which is AST3, that showed improvement over co-injection with the isotype control, but actually even outperformed the unmodified antibody with FC gamma receptor blocking antibody. So this was our winner that we took then to further experiments. So right now we have a DNA binding antibody with FC modifications that seems to decrease the clearance of injected double-stranded DNA. But what does it do for us in terms of tumor detection on endogenous DNA? And so similar to the nanoparticle work, we turned to a long metastasis model where we have a panel targeting the mutations in this luciferase CT26 cell line, and we're able to detect these mutations in the plasma of these mice. And we injected these mice with either unrelated IgG2A control antibody or our engineered variant, and then sampled blood 2 hours later. And what we were excited to see is that with the engineered antibody, we can get over an order of magnitude improvement in detection of cancer mutations in plasma. And consistent with this, we were also able to detect a larger percentage of the mutations that we were tracking. So with the control antibody, we could only detect about 12% of the mutations that we had built a panel for, whereas with this priming agent, this contrast agent, we could detect 75, 80%. So now, just to bring everything back to the beginning, right now we've shared with you 2 different stories for how we can develop molecular contrast agents for liquid biopsies. In the first part, we talked about a nanoparticle-based contrast agent that acts on the cells responsible for cell-free DNA clearance and allows us to recover more cell-free DNA and detect more tumor mutations in plasma. Um, for us, the next steps in this direction are to further optimize the nanoparticle formulation and further reduce the dose and understand the generalizability of this approach to other liquid biopsy analytes. And then on the DNA binder contrast agent side, we've shown you how a DNA binder can protect cell-free DNA itself from clearance. And the next questions for us in this avenue is how we can improve on the observed effect using alternative protein scaffolds or higher affinity antibodies, and whether it would be possible to specifically enrich for tumor DNA in plasma rather than all DNA. And of course, for both of these approaches, questions regarding clinical translation, dosing, tolerability will be critical. And just taking a step back, I think this approach also introduces really a concept of molecular contrast agents for endogenous biomarkers in vivo. So an approach in which you can increase the amount of an endogenous biomarker to improve sensitivity in similar ways to how we already inject gadolinium contrast agents to make a brain tumor visible. We envision that these approaches could help overcome the sensitivity issues that are limiting the liquid biopsy field currently. And we envision that this work could really increase the clinical utility of liquid biopsies, perhaps spark interest to further develop contrast agents for other circulating biomarkers, and be used in other disease areas beyond oncology as well. And with that, this has been a huge collaborative effort between the Bhatia Lab, Adalsteinsson Lab, and the Love Lab, supported by many folks. So just a big thank you to everyone, including all our supporters. And with that, we're happy to take any questions. Yes.
Hi, thanks, guys. This is super exciting work. I'm really impressed every time I see it. I was wondering, and you set it up in the last slide, do you know if your sensitivity is higher than imaging techniques? So can you catch a tumor with cell-free DNA before we see it in a scan?
Yeah, we haven't really benchmarked against any imaging modalities, so I think that's high in the to-do list. Um, and to be honest, like with some of these, I think one of the challenge is that some of these models are not really physiologically shedding. Like we're really early days in terms of how these tumors seed and then start shedding. So I'm not sure they're quite clinically relevant just yet. So I would be a bit cautious in sort of interpreting how imaging would benchmark to this ctDNA analysis. But yeah, I think this is something that for the next tumor brain cohort, we will for sure try to benchmark to.
I've got the mic, so I'm gonna go. Can you talk about what you know about the turnover rate at this Kupfer cell saturation? Like how long of a window does that buy you when you pre-administer with the liposomes?
Yeah, to the extent that we've done this, really, is like we've tried either waiting an hour or waiting half an hour, and we see sort of comparable effects. We haven't really tried beyond that. We've also done sort of in vivo imaging to see how long we keep the liver busy, essentially, like how does the liver occupancy look in vivo, and we know these liposomes essentially like accumulate immediately, pretty much like as soon as you start imaging the mice, they're there. And then depending on the mice, on the dose, sorry, they either stay there and then peak at like an hour or like even like 3 to 4 hours out, but then they rapidly get cleared. So that's sort of like the kinetics we've observed so far. Obviously, like depending on pegylation and formulation, this will like drastically change. So I think there's a lot of opportunity there in sort of exploring different formulations and seeing how this affects kinetics of cell-free DNA recovery.
Great talk. It was super fascinating. I have one question for each, but they'll be really fast. So for the nanoparticle contrast agent, do you guys think that there's any possibility that part of the mechanism of action is like histones or DNA interactions with your nanoparticle, and that just facilitating some degree of stability? And then on the DNA binding contrast agent side, on the gel shift assay, you had shown like it really binds well to, double strand DNA alone, but not really with high affinity towards the ones bound by nucleosomes. Does that introduce some set of bias from a sequence point of view? And could you see that like with predictions on where nucleosomes typically bind?
Very good questions. For the sort of interactions between nanoparticles and cell-free DNA, this is something we explored early on, but we haven't really like thoroughly characterized. Um, we didn't observe, like one of the functional tests we did was just like look whether when we co-incubate the particles with cell-free DNA, whether we had nuclease protection. So, under the assumption that there would be some steric hindrance and we would protect the DNA if it was binding. We didn't observe that to be the case. But I think there's still room to sort of explore that avenue. One thing is that we wouldn't really, if we're really designing these particles to target and go to Kupffer cells, we wouldn't necessarily want it to bind. Otherwise, we would just be funneling it to Kupffer cells. But there might be some interactions we're not aware of happening.
And then on the antibody, so really good question. What we have looked at sort of the sequence specificity, and it's actually part of ongoing work. But to your point, one of the things that we've noticed, which I didn't have time to really delve into here, is that it seems that we get a preferential enrichment of DNase hypersensitive sites in the mouse genome, which are sites that would not be occupied by nucleosomes. So I think there's probably some preference in that regard. There's also some preferences in terms of sequence contents, such as GC content that we're exploring. And then, yeah, on the gel shift assay, one of the interesting things on that assay was at the highest concentration, both bands, both the nucleosome and the free double-stranded DNA bands nearly disappear. So we do think it's at least interacting with both forms pretty well at the highest concentration, but it may be that it's actually displacing the DNA from the histone octamer.
Thank you. Yeah, that makes sense. More questions?
So with the liposome treatment, have you looked to see if there are other interesting things that you accumulate in the blood, like exosomes, or I don't even know what else, but exosomes came to mind as something else that might be interesting information.
Yeah, that's a very good question. We're actively trying to figure that out. We think exosomes are a very obvious analyte that could be being enriched for. So that's in the pipeline. We don't have any data to suggest that just yet.
I understand it's very big. You're still on the very basic level of understanding. How long is the? I guess my question is, how permanent are this going to be? Your antibodies are, they're going to be. How long they're going to be keeping the DNA material in the blood? Will it be cleared at all at some point?
Yeah, that's a great question. So the reason why we did the sampling at 2 hours was actually we started noticing a decrease after 2 hours. So I think 2 hours is really the peak time that we see. And we see the vast majority is cleared. We've done some pharmacokinetic studies on the antibody itself and see that the vast majority is cleared within about 48 hours. So I think up to 24 to 48 hours is probably where most of them stick around, and then after that they're clear.
All right, thank you very much.
Thank you.